Let us continue our discussion on human health and disease, an important chapter from your class 12th NCERT biology syllabus for NEET entrance exam. And we will talk about AIDS in this video. So AIDS stands for acquired. So let us see what does it mean. So it means acquired immunodeficiency syndrome. So this is acquired immunodeficiency syndrome. Now let us try to understand. So this disorder is actually an acquired disorder. So you can contrast it with a disorder which is called as congenital, that is present since birth. So it is not present since birth. Yes, it can be transmitted across the placenta. It can be transmitted across the placenta by an HIV positive mother to the fetus, no doubt about it. But still we will say that it is an acquired disorder, not a congenital disorder. And if you look at this term, this is immunodeficiency. So the immunity is drastically reduced, significantly reduced. So this is not zero immunity. This is immunodeficiency, which is significantly reduced, and therefore the person may have lot of infections, and therefore the life may not be possible with all these type of infections very readily happening in the patients suffering from AIDS. And if you look at this word, this is syndrome. And this syndrome means that it has a constellation of signs and symptoms. So it is not only a few signs and symptoms, there are a huge number of signs and symptoms, and such type of diseases are actually called as a syndrome. So if you are looking at the timeline, the first case of acquired immunodeficiency syndrome came into light in 1980-81. This is, we are talking about Los Angeles, USA. And in another 40 years or so, if we are talking about 2020, 2021, so it's an approximately period of 40 years. A lot of research has gone into understanding the various aspects of AIDS, and still though there is no cure, but this is now basically a chronic type of disease, and if the person takes the medicines actually meticulously, then is expected to live nearly normal lifespan, and that too in a healthy manner. So this is what has happened with the development of the science. Now, what causes it? So the next question would be the cause. What is the agent? So the agent is a virus. This virus is called as HIV, and this means human immunodeficiency virus, and this virus is a retrovirus. So this virus is a retrovirus. So retrovirus has a capsid. So I am making not HIV but a generalized retrovirus, because your NCERT gives the life cycle of a generalized retrovirus. This is what we are going to discuss. So it has a capsid, and in the core will have single-stranded RNA as its genetic material, and will have an enzyme which is called as reverse transcriptase, and also has an enzyme which is called as integrase. So we are looking at a retrovirus, and this retrovirus we are saying that has single-stranded RNA as its genetic material. It will have an enzyme which is called as reverse transcriptase. I have already discussed about reverse transcription in the chapter on molecular basis of inheritance, and it also has an enzyme which is called as an integrase enzyme. So going by the diagram that is given in your NCERT, so which tells you about the life cycle of a generalized retrovirus, it says that this virus will go and bind to the receptor that is present on the animal cell. So this is an animal cell. The retrovirus comes and binds to the receptor that is present on the cell and injects its single-stranded RNA into the cell. So let us try to keep the color scheme intact, and let us say that this single-stranded RNA now enters the cell, and so does the enzyme reverse transcriptase as well as the integrase enzyme. Now if this retrovirus has to infect an animal cell, and an animal cell is a eukaryotic cell, and the genetic material of the eukaryotic cell is double-stranded DNA, and if at all it has to take over the metabolic machinery of this cell in order to replicate itself inside the animal cell, it must make this complementary DNA on the template of RNA. So the DNA that is formed on the template of RNA, this would be reverse transcription with the aid of an enzyme which is called as reverse transcriptase. This type of DNA would be called as complementary DNA. So the next thing that NCERT says is that what happens is that the viral DNA is formed. So the viral DNA gets formed. So this happens, the viral DNA is formed, and then if you watch your NCERT, it says that this goes and gets integrated into the genome of the host. So what happens is that this would be the DNA of the host animal cell, and what we are saying is that this complementary DNA that is produced because of reverse transcription will go and will get integrated into the genome of the host. This is how it takes control of the host genome. And then what happens is that further, now this DNA will be transcribed and new viral RNA molecules will be formed. So we are saying that again the viral RNA molecules are formed. Now you can understand, now this is a forward flow of the genetic information, and this DNA will get transcribed and the viral RNA molecules will be formed, and these viral RNA molecules will be containing the information to make the capsids or the protein coats, and then they are synthesized with the help of getting these RNA molecules translated, and we are saying that these capsids will be formed. And finally this is very important for our biotechnology as well. Now this is an important concept that I am slightly deviating from the diagram given in NCERT, just to tell you something that will come to our use in biotechnology, that if a particular RNA molecule, this viral RNA molecule, has a particular sequence which is called as a packaging sequence, if it is present in this molecule, it will get packaged into the capsids, and the new virion or virus particles will be formed. And then NCERT says that these will be able to escape the cell. So they go and come out of the cell, and there is a note given in NCERT which says that it is not necessary that the cell will be killed. The cell may actually survive, and the virus may be able to replicate itself inside the cell for a very long period of time, and these new viral particles are actually able to now infect the new host cells. I hope this diagram is a very important diagram. Try to see all the steps given in NCERT. A direct question may come where they can actually label them as A, B, C, D, and they can ask you what is happening at what step. So this is what is happening in the life cycle of a generalized retrovirus. This is a diagram. Actually, I will advise you to pause the video right now, go to your NCERT, open the page and see the diagram that is given in the NCERT regarding the generalized life cycle of a retrovirus now. And when you have done it, then start the video again and let us continue the discussion further. Now, this HIV again is a retrovirus, which means now we know that what exactly is a retrovirus. And about HIV, this is very important to understand that it has actually two molecules of single-stranded RNA. Now please make a note of this. So HIV has two different molecules of single-stranded RNA. Now it is not a double-stranded RNA. These are two molecules of single-stranded RNA. Obviously it will be having reverse transcriptase, it will be having integrase as well. So this has two molecules of RNA. You know that RNA viruses mutate very fast. There is a term which is called as antigenic drift. There is a term which is called as antigenic shifting. You must have heard about these terms with respect to the coronavirus. And then there are problems in dealing with the new mutant variants of the viruses. So it can mutate very fast. That may actually explain one of the probable reason why, in spite of so much research and development, an effective vaccine to prevent HIV infection is not yet possible, and there is no cure yet of AIDS. I hope this thing is now clear. Finally, one more thing to understand would be that it is actually enveloped it is actually enveloped. The envelope is of the source of the envelope is actually the host cell. And there is an important thing, if you are able to remember, what is called as a glycoprotein GP120 that is present on the surface of HIV. So this is how we will try to tell you what is the structure of HIV looks like. I hope this thing is now absolutely clear. Obviously, the next question would be that how is it transmitted? How is it transmitted? So that means it jumped to the human beings, but it was actually infecting the apes and then got mutated and got the capability of infecting the humans and jump onto the human side. And now among humans, it is transmitted. This is the mode of transmission. The number one transmission mode would be a sexual contact. So unprotected sexual contact would be the number one reason for the transmission of HIV amongst humans. Homosexuality increases the risk of transmission of HIV, but because the number of heterosexuals is much more than homosexuals, so that remains the number one cause. The second thing would be the transfusion of contaminated blood. Or not, it is not necessary that it has to be complete blood. It can be blood products as well. So this is another method by which HIV gets transmitted, and this is why it is very, very common in IV drug abusers. So intravenous drug abusers can get infected with HIV more commonly. And the third mechanism would be what is called as the vertical transmission, and another name for vertical would be transplacental. So just now I told you that an infected mother can pass on the condition to the developing fetus through the placenta. So this is transplacental. Obviously, let us add here that yes, lactation is also known to cause the transmission. So these are the modes of transmission of HIV. A very important point, NCERT says here that it is not transmitted by casual handshaking, hugging, etc. And therefore there is no need for any stigma associated with this condition. So in the society, people who are HIV positive are actually looked down upon. There is a huge stigma associated with it, but this is the nature of the science, that it busts the myths. And now you know as a student of science that these are the only mechanisms by which it is going to be transmitted. Now the next thing would be what happens when HIV enters into the human body. So when HIV enters into the human body, the person becomes HIV positive. The person becomes HIV positive and will develop a viral prodrome. So all viral infections are associated with certain signs and symptoms which are common to many viruses. Same thing will happen here. There may be fever, there may be myalgia, there may be a sore throat, etc. These type of things, and then the things will get normal. And this is what is called as the first stage of HIV infection. The person is HIV positive, but there is no way that this can be suspected. And this is a dangerous period because the person is HIV positive and can spread the infection to other people. Now what happens is that in initial stages, it actually enters what are called as macrophages. This is what your NCERT says. Now important point, and you know that macrophages are actually cells involved in immunity. So you know about their very important role played in innate immune response. And what happens is that these macrophages are used as factories by HIV. I am borrowing the word from your NCERT, and they keep on replicating it. They do not actually kill them, and they keep on replicating inside the cell. They increase their number. So suddenly what is going to happen is that the viral load in the body is going to increase. And when there is sufficient viral load, what happens is that they will enter what are called as T helper cells. And T helper cells, as you know, are also called as CD4 T lymphocytes, and they are very, very critical for an adequate innate as well as adaptive immune response, both humoral as well as cell mediated. So both humoral as well as cell mediated immunity, if they are to be adequate, they depend on certain chemicals secreted by the T helper cell. And And what happens is that this is the cell that it actually destroys. So it infects and selectively destroys the T helper cells, and then the T helper cell count will actually come down, and it is going to reduce the immune power of the infected person. Now this is very easy to understand. So what happens meanwhile is that the person as I told you may actually have simple a viral prodrome, and then may actually develop what is called as a lymphadenopathy. So the lymph nodes may be enlarged, and then can actually have now you can understand that the immunity is getting lower. The person may have unexplained diarrhea, and diarrhea is loose motion. Normally all diarrhea cases, if you can explain them what is the cause, they should be very well treated within 1 to 2 weeks. But somebody is having a continued diarrhea and you are not finding any cause, this is an unexplained diarrhea. And similarly there would be significant weight loss. So this is significant weight loss. I am not merely saying weight loss, I am saying significant weight loss. Incidentally, HIV in the beginning used to be called as slim disease because of the significant weight loss that we are talking about. But still it is not AIDS. The person is HIV positive. This is an important point to understand. HIV positive does not mean AIDS. Now what happens in AIDS is that the T helper cell count actually becomes significantly low, and this leads to a decrease in both humoral as well as cell mediated immunity. Obviously the innate immune response will also be adversely affected, and the now person is not having much of a defense against the microbes that normally were not able to cause infection in them because of the presence of adequate immunity. So I am saying it again. So there are you are surrounded by huge number of microbes. They are not able to infect you because of your immune system. But if the immune system problem is there and it is not functioning properly, then these microbes will get an opportunity to actually infect the person who is suffering from HIV infection. I hope you are getting the point. And therefore, in addition to this, the person starts developing what are called as opportunistic infection. Can you imagine? Can you understand what a good name they have actually given to these type of infections? They are called as opportunistic infections. Just now I told you that normally these microbes will not be able to cause infection, but now they get an opportunity to do so. And now with the development of the opportunistic infection, we will say that the person has what is called as acquired immunodeficiency syndrome. Now this person would not be simply HIV positive, but will be suffering from AIDS. And your NCERT talks about Mycobacterium tuberculosis. This is the most common opportunistic infection associated with HIV infection in AIDS patients. The second thing would be toxoplasma, and these are the two things that are given in your NCERT, but I will tell you two more. One is pneumocystis carinii pneumonia. Pneumocystis carinii pneumonia. Now it is actually called as pneumocystis cerevisiae. So this is pneumocystis carinii pneumonia, and the second is a mesodermal cancer which is called as Kaposi sarcoma. So just going outside NCERT, please try to remember if possible these two names. These have been very important, and a question, if ever it comes outside the NCERT, this is the one that they can actually ask. So this is what is the pathogenesis of AIDS, and you know about the clinical features. All these clinical features will be because of these infections. We have already told about lymphadenopathy. I have already told you about unexplained diarrhea, significant weight loss, and all the signs and symptoms associated with these various infections will be present, and therefore you can understand the lifespan will be curtailed. It is not easy that to actually treat the opportunistic infections if they are not diagnosed in time. Now after this there is an important thing. The next important question is that, okay, now we know that what is AIDS. How do you diagnose it? how do you diagnose AIDS? So the AIDS can be diagnosed. Number A, there is a preliminary test which is called as the screening test. This screening test is called as ELISA, and we will be talking about ELISA in chapters on biotechnology. So this is a screening test for AIDS, HIV detection, and those who come positive in ELISA are subjected to confirmatory test, which can be a Western blot. We will be talking about all these tests in chapters on biotechnology, the molecular diagnosis subsection, and you can always think of polymerase chain reaction. So RT-PCR, all these type of things can be done to confirm that the person is suffering from AIDS. Now, after diagnosis, obviously the next thing would be treatment. So unfortunately, when we talk about treatment, we say that it has no cure. So it has no cure, but still you treat the person. So still you treat the person, and the idea of providing treatment to these people are that somehow the viral load of the body should be reduced, and somehow you interfere with the replication of the virus inside the body so that the viral load is actually reduced. And we give what is called as highly active or effective antiretroviral therapy. So this is called as HAART, and the basic cornerstone that the drugs that are used in HAART are antiretroviral drugs, and these are the drugs that actually inhibit the reverse transcriptase enzyme. So you can easily understand the importance of the reverse transcriptase, and the drugs that are used to interfere with the replication of the virus actually are reverse transcriptase inhibitors. These are the mainstay. As I told you, a lot has been done, a lot of research. There are some very amazing drugs available, and persons who are actually HIV positive can be, if treated in time and treated properly, as I told you, can have a near normal lifespan, and that too with quality. Now, if there is no cure of a disease, what would be better? The answer is prevention. And how do you prevent the disease? By spreading awareness. So you know about the mode of transmission, get the people aware about that, how this is transmitted. You tell the people about the safe sexual practices, the use of barrier contraceptives, safe transfusion practices, antenatal screening of the mother, because if the pregnant female is identified actually that she is suffering from HIV, and you start HAART well in time, then in most of the cases the child that is born is actually not HIV positive. That is a remarkable thing. Obviously, the lady is advised not to lactate the child after the birth. So all these things, you know about the mode of transmission, immediately you get an idea that how do you prevent it, and getting people educated, aware about this thing is very, very important. And for spreading awareness, obviously, you will require a nodal agency, and this nodal agency is National AIDS Control Organization, and the headquarter is in New Delhi. And you know about the symbol, it is actually a red ribbon. So this type of symbol, you know about AIDS. This is about spreading the awareness regarding the AIDS.